Hello friends, and welcome to our video lecture reviewing digestion and metabolism biochemistry. Our essential understanding for that topic 6.1 digestion, the structure of the wall of the small intestine allows it to move, digest, and absorb food. Our objectives for this crazy fast review are going to be to look at metabolism and biochemistry. We're going to talk about the structure and function of digestive system organs with a big focus on the small intestine, since of course that is what our essential understanding is all about. So remember that biomolecules are what are needed for life processes. There are 4 major classes of biomolecules: carbs, lipids, proteins, and nucleic acids. We're going to talk about macromolecules and monomers of each. So some examples of macromolecules of carbohydrates: of course, we have glycogen, and we have starch, which is composed of amylose and amylopectin, and we have cellulose. Glycogen and starch are all composed of alpha glucose monomers. Mono is 1, those are those tiny little pieces. Cellulose is made of beta glucose. We mammals are good at making enzymes that break down polymers of alpha glucose. We're not so good at breaking down cellulose, but at least we've got some fiber. Carbohydrates are going to have carbon, hydrogen, and oxygen. We're going to have equal-ish, not exact, but close numbers of carbon and oxygen. We're going to have just about twice as many hydrogens as we have oxygens. Lipids are also carbons, hydrogens, and oxygens. These guys aren't exactly good at fitting into that macromolecule monomer categorization, but we can talk about things like triglycerides are a little bit bigger of lipids. They are the combination of lots of fatty acids. Fatty acids would be some of the monomers-ish of lipids. Proteins are going to be carbon, hydrogen, oxygen. We're going to add in some nitrogen always, and then sometimes some sulfur. Protein macromolecules, basically proteins, right? We can also refer to those as polypeptides if we're talking just about like the chains of amino acids. Amino acids are the monomers of proteins. Those amino acids are held together by peptide bonds in those polypeptide chains. Last but not least, we're going to talk about nucleic acids. Nucleic acids, carbon, hydrogen, and oxygen always. Also some nitrogen, and then we're going to throw in some phosphorus. Nucleic acids consist of DNA, RNA, and then those monomers are going to be nucleotides, A's, T's, C's, G's, and U's. So quick review about metabolism. How do we make and break down these biomolecules? Metabolism is the sum of all enzyme catalyzed reactions in a biological system. And metabolism comes in 2 kinds. There is anabolism and catabolism. Anabolism, we are building things up. We call those kinds of chemical reactions condensation reactions. Sometimes they're also referred to as dehydration synthesis. In IB world, we call them condensation. In AP world, we call them dehydration synthesis reactions. Basically, what happens is we take 2 little monomers or other smaller things. We're going to combine them by pulling water out. So this is why we call it dehydration synthesis or condensation. We're going to pull some water out, a hydrogen from one guy, a hydroxide, hydroxyl group, OH, from another guy, and then that O and the C that's left here, they're going to bind to each other. There it is right there. Actually, I guess right there technically. And we're going to call this kind of a bond between 2 monomers of carbohydrate. It's going to be a glycosidic bond. And glycosidic bonds can come in a couple different forms. We can have 1,4, which is what we have between these 2 guys. If we have branches, then we would actually have 1,6. So amylose is one of those carbohydrates that has only 1,4 glycosidic bonds, whereas amylopectin and glycogen would have 1,4 and 1,6, those branches that kind of pop up sideways. Anabolism, these condensation reactions, is going to require some energy. We're going to add some energy to the system. It's going to look kind of like this graph here, endergonic reactions, where we have more energy getting pulled up when we make our products. Products have more energy than our reactants had. We're going to have to add this energy. Enzymes are super cool. They are going to reduce activation energy here without an enzyme, only here with the enzyme. But this is endergonic anabolism. We are adding some free energy. It's going to entail a decrease of entropy. Things are a little bit less chaotic because the molecules are bigger, but it's actually hard for us to hold big molecules together. So catabolism is the opposite. We're going to break things down. I remember this as C for cut. So catabolism, we're going to cut big molecules into smaller molecules. Anabolism, we are building things up bigger, like anabolic steroids make bigger muscles, but they're bad. Don't do them. Anyhow, back to catabolism. So catabolism, we are going to cut some big molecules down into smaller molecules. It's like decomposition. We're going to use these hydrolysis reactions. In these ones, we're actually going to add some water. One of the H's goes onto one side, one of the OH's goes onto the other side, and we end up with OH's and H from that water, and it's going to split. In this case, we have a dipeptide breaking apart into 2 amino acids. We're breaking that peptide bond through hydrolysis. Catabolism, this is usually a little bit more spontaneous. It's going to be exergonic. We're going to have more energy in the reactants than we have in the products. And even though we have to add some energy, that activation energy, the final amount of energy in the products is a little bit lower. That's going to be exergonic, a release of free energy, which usually means that it's going to be spontaneous. Let's take a look at lipids. So lipids are used for long-term energy storage. We use them as insulators. Fat, adipose tissue is a great insulator. Hormones can also sometimes be lipids, so we do also have some protein hormones. Examples of lipid hormones, actually I've got some listed down here. Testosterone, estrogen, progesterone are all hormones that are lipid-based. They, as I said before, lots of carbon and hydrogen, just a little bit of oxygen. So those carbohydrates, remember we talked about CH and O, equal-ish, not exactly, but close to the same numbers of carbon and oxygen, and then hydrogen will be twice as much oxygen. That's very different here in the lipids. We're going to have so much hydrogen and a lot of carbon, just a tiny, tiny little bit of oxygen. They are hydrophobic, which means that they are repelled by water. They do not dissolve well in water at all. An example of a lipid is a triglyceride. This is an example of a triglyceride. We have a glycerol backbone here. We have 1, 2, 3 ester bonds holding these fatty acid chains to the glycerol backbone here. And because there's 1, 2, 3 fatty acid chains, we call it a triglyceride, glycerol and 3 fatty acids. We can also have phospholipids. Phospholipids are going to have a phosphate head and 1, 2 fatty acid chains. And then steroids are not really combinations of fatty acid chains at all. Instead, they mostly look like this, cholesterol. And then, as I mentioned earlier, testosterone, estrogen, progesterone, these are all steroid-based hormones that look a whole lot like cholesterol. So lipids, these fatty acids come in a couple different shapes and forms. They can be saturated, which means that there are no double bonds between the carbons and the hydrogens. There's this double bond here between the carbon and the oxygen. We need that to make it the acid part. This piece of the molecule is known as a carboxyl group, and that carboxyl group makes this whole thing a carboxylic acid. So this part makes it an acid. This is the part that makes it kind of fatty. This is saturated because in this part of the molecule there are 0 double bonds between carbons, 0 carbon-carbon double bonds, which means that we can saturate this molecule with the maximum number of hydrogen atoms. These guys tend to be solid at room temperature. Things like butter and bacon grease taste really good, but they're not super great for our blood vessels. They tend to stick to the insides. And the word that I never say very well, arteriosclerosis, can happen, that hardening of the arteries, because we have these saturated fats sticking to the insides of our arteries, which is going to reduce the amount of space that we have, which is going to increase blood pressure and really could lead to coronary heart disease and heart attacks. Bad, but also, I mean, moderation, right? And then sometimes better for us are unsaturated fatty acids, where we do have at least 1 double bond between carbons. So carbon-carbon double bonds. Again, this is part of that carboxylic acid group. Don't worry about that double bond. It has to be there. If we have only one double bond between carbons in the fatty acid chain, and that carbon chain going away from the carboxyl group, then we're going to have a monounsaturated, just one double bond. Polyunsaturated, we can have multiple double bonds. Here we have one double bond, two double bonds. This is going to be polyunsaturated. These unsaturated fatty acid chains can be either cis or trans. Cis is same, and trans is opposite. So if I have the hydrogens that remain from where that double bond are, if they're on the opposite sides, then we're going to have a trans fatty acid, trans unsaturated fatty acid. These are actually not that great for us because they're still kind of straight-ish, and so they tend to stack the insides of our blood vessels, much like those saturated fatty acids. However, these cis unsaturated fatty acids are so good. I mean, they're really better for us than the saturated ones are. But of course, too much of anything is not going to be good. So don't get excited and drink bottles of olive oil. That's also not good. But what happens is these double bonds with the hydrogens on the same side of the carbon-carbon double bond, this is going to lead to this little bit of an empty space, which means that the molecule will fold in a little bit. These kinks in the molecule are going to prevent them from stacking together super, super tightly, which means that they're more likely to be a liquid at room temperature. Liquid at room temperature, more oils, less fats, and these are better fats for us. But again, don't go drink a whole bottle of olive oil. That's not a good idea. All right. Let's talk about our next biomolecule nucleic acids. They are DNAs and RNAs. Remember that RNA has uracil, DNA has thymine, DNA is double-stranded, RNA is single-stranded. RNA is going to have ribose as its sugar, and then DNA has deoxyribose as its sugar in that sugar phosphate backbone. So ribose, phosphate, ribose, phosphate, ribose, phosphate, whereas here we have deoxyribose phosphate, deoxyribose phosphate. Oh my gosh, say that 5 times fast. Nucleic acids, the structure of DNA was elucidated, I think that's a word, by these guys, Watson and Crick, and they won a Nobel Prize for it. But we all know the very salty story about Rosalind Franklin. This was the photograph, the image, X-ray crystallography image that she made that was taken without her permission to Watson and Crick, and they used that information to figure out the DNA was a double helix. They got a Nobel Prize. She didn't. It's a salty story. Remember Rosalind Franklin. Seriously, girl boss, right? All right. Proteins. So proteins are formed from amino acids. This is a generalized amino acid. All amino acids have an amino group. They also are going to have that carboxyl group. This is the part that makes it the acid. This is the part that makes it an amino acid. And that central carbon in the middle is going to be bound to a hydrogen, and then this R group. R is the side group or the radical group. This is the thing that's going to make each amino acid different from all the other ones. So different R's, but the rest of the structure of the amino acid is the same for all of them. Amino acids are held together by peptide bonds, which is going to lead to the formation of polypeptides. How do we make our polypeptides, our proteins? Well, we start with that DNA code. Genes are sequences of DNA that code for that protein. We do that by going through transcription to make messenger RNA. That messenger RNA is going to head out to a ribosome. The ribosomes are going to use that mRNA transcript to make, through translation, a sequence of amino acids that is that polypeptide. Proteome is a vocab word that is the sum of all the proteins produced by a cell or a tissue or an organism, depending on what we're looking at. We can figure out what those proteins are by looking at gel electrophoresis, so we can sort out protein by sizes, sending them through electrophoresis, just like we do with DNA. If we wanted to talk about some DNA, there's also this word genome, which sounds a lot like proteome. Proteome is the sum of all the proteins. Genome is the sum of all the genes in a cell or an organism, whatever it is that we're looking at. So different examples of proteins. Proteins can be enzymes. Examples are rubisco and amylase. Rubisco is involved in carbon fixation in photosynthesis. Amylase, if it ends in an ace, it's an enzyme. Amylase breaks down amylose into maltose, which is then broken down by maltase into alpha glucose. Proteins can be hormones like insulin. Insulin is a hormone released by beta cells of the pancreas when our blood glucose levels are too high. It's going to cause that glucose to get bound into glycogen, which is stored either in our muscles or our liver, reducing that blood glucose level. Antibodies are also proteins. Antibodies are made by plasma cells, which are differentiated B cells. Sometimes we can also call these antibodies immunoglobulins. Oh, that's sometimes a hard word for me to say. Immunoglobulins. These guys are going to bind to specific antigens of very specific pathogens in order to prevent, slow down, do something to that pathogen so that we don't get so sick, or to help us get better from being sick. We have some pigments. An example of a pigment protein is chlorophyll. Chlorophyll is an amazing protein complex that makes plants look green to our eyeballs and also helps with light absorption and electron excitement. And then, of course, we have lots of proteins involved in structure. Collagen, keratin in us animals helps hold our skin together, makes for pretty hairdos. And then, of course, spider silk is involved, a protein involved in webs of friendship, of course. And on that note, we're going to go ahead and call our review of metabolism and biochemistry done. We're going to flip over to digestion. So topic 6, human physiology. Topic 6.1, digestion. The structure of the wall of the small intestine allows it to move. We're going to talk about how food gets moved, digested, how do we digest that food, and how do we absorb food. The small intestine is incredibly important for all of these things. This image of small intestine, this little section of small intestine, is way more detailed than we need. So I'm going to just focus on the stuff that we do need to know. So serosa is going to be the very outermost skin epithelial piece of the small intestine. Just inside that, we're going to have the muscles. So we have longitudinal muscles and we have circular muscles. Longitudinal muscles go down the length of the small intestine. Circular muscles wrap around in circles around the small intestine. We have the submucosa, which is lots and lots of connective tissue just inside the mucosa. The mucosa is where we're going to find the villi and then microvilli that aid in absorption. Epithelium, again here, just that outermost covering, those epithelial cells of the small intestine have those microvilli, tiny little projections on the cells of the villi, which are these finger-like structures. This is the lumen of the small intestine, and food is going to move through there. Good? All right. So we're going to test ourselves super quick and see if we remember all those pieces and parts. So here's the epithelium, that very outermost layer of the small intestine. Just inside we have this guy. It's called the serosa. So we have serosa. Just inside the serosa, there's some muscle. We have longitudinal muscle, and then we have circular muscle. I remember that circular is closer to the center of the small intestine. Just inside the muscular layers, we have the submucosa, lots of connective tissue just holding everything together. And then up here is the mucosa. The mucosa is where we have the villi. The villi are those big finger-like projections. We also have microvilli. Microvilli are tiny little projections on the cells, the epithelial cells of the villi. I'm going to add in here, we also have some blood vessels, so capillaries, that are going to circulate through the villi, capture those nutrients, take them to the liver first. The liver will process all these nutrients that are getting absorbed and then decide where they go. We also have this guy here is the lacteal. Lacteal is going to transport fats through the lymphatic system. Those fats clog up our blood vessels really easily, and so it's safer for us to send fats through the lacteal to get processed. The other nutrients are going to go through these capillaries to the liver to get processed. And even though the small intestine is the organ that we're going to talk about the most, the other ones are also super cool and important. So we're going to take just a quick minute to look at these other guys. We've got teeth and mouth. This amazing tube is called the esophagus. Sometimes in IB world, we throw an O in front of it. Peristalsis happens here in the esophagus. Peristalsis is that sequential contraction of muscles that allows food, and we call the food that's getting sent down the esophagus the bolus. So the bolus is going to travel through the esophagus via peristalsis. From the esophagus, we're going to send that food into the stomach. The stomach is pretty cool. It has a really low pH because it's highly acidic. All that stomach acid that we are producing, that stomach acid has a couple jobs. It's going to help with protein digestion. And then it's also going to help to kill bacteria. So that crazy high acidity, low pH helps us kill a bunch of bacteria that might be in our food. We only start to digest proteins, which is weird because we're usually like, oh, the stomach is where we digest all our food. In fact, we only begin protein digestion in the stomach. Everything else is digested in this amazing organ, the small intestine. Food is going to go from the from the stomach into the small intestine. We are also going to have this super cool organ that's hiding back here is the pancreas. The pancreas is going to make lots and lots of enzymes. Those enzymes are going to get dumped into the small intestine, and then digestion will occur there in the small intestine using those enzymes from the pancreas. The other thing that gets dumped into the small intestine from the pancreas is bicarbonate. That bicarbonate is going to help to neutralize the stomach acid because even though the stomach is built for a very acidic environment, the small intestine is not. So bicarbonate from the pancreas, let me erase all my scribbles. It's maybe hard to kind of see. There it is, maybe easier to see now. Pancreas hiding back here. Pancreas is also going to make that bicarbonate that is going to neutralize stomach acid, go back to a slightly basic, just a little bit more basic than neutral environment in the small intestine. The other cool thing that we've got going on here, this is the gallbladder. The gallbladder is going to receive bile. Bile from the liver is going to go into the gallbladder, and then it's also released into the small intestine. That bile is going to help to emulsify fats. So fats we know are hydrophobic. They do not like to dissolve in water. And if they're all clumped up like this, it's hard for our enzymes to get to them and break them down. So what that bile does is it just helps to open up those fat molecules, emulsify, just open them up, make it easier for our enzymes to get to them because they're not all clumped up together. That's called emulsification. And it's done by bile from the liver stored in the gallbladder. Small intestine, again, all of that digestion. So we're going to finish digesting our proteins. We're going to digest starches. We're going to digest nucleic acids. We're going to digest, what did I forget? Carbohydrates, lipids, nucleic acids. We're going to finish digestion of proteins. We're also going to continue doing that peristalsis that we did up in the esophagus. We're going to have some peristalsis that's moving the food through the small intestine so that it can eventually get to the large intestine. Lots of absorption is happening here in the small intestine as well. Those villi and microvilli have lots of ways to grab onto nutrients as it travels through the small intestine. Large intestine, also known as the colon, finally, is going to absorb a lot of water. So we're going to keep a lot of the water from our food. We don't necessarily want to poop out all of our water unless we're super sick and we're trying to flush things out. That's called diarrhea. It's kind of gross. But most of the time, the large intestine is going to absorb that water and get us to a healthy osmolarity. Good. All right, so a lot of these words that I just said on that previous slide, so mouth, teeth, tongue, we have that mechanical digestion. Salivary glands, our spit glands, are going to make a little bit of amylase. We're going to start to break down starch in the mouth. Esophagus is going to move that bolus, that chunk of food that we swallow, down to the stomach through that peristalsis, that sequential muscle contraction. The stomach is where we have some mechanical, we have lots of churning and stirring of stuff. And then we're also going to start to chemically digest just our proteins. So pepsin is a protease. It's an enzyme that breaks down proteins. We have that hydrochloric acid that helps to make that pepsin work really well. It also helps to kill bacteria that might be in our food. Because we do have such a low pH, because the stomach is so acidic, its stomach wall. The stomach wall is lined with lots of mucus to protect us. If that mucus breaks down a little bit, then we end up with what's called an ulcer, and that's bad. Peristalsis, just a quick look at that in more detail. It's the sequential or alternating contraction of circular and longitudinal muscles through the gut, esophagus, and intestines. Is going to move that food through the digestive tract. So we're going to squeeze this way, and then we're going to squeeze that way, and then we're going to squeeze this way, and then we're going to squeeze that way, and it's going to cause that food to just shimmy on down the esophagus or the small intestine or the large intestine. Small intestine, this is where we finish up digestion of all of our nutrients. We're going to absorb all the nutrients. And of course, as I mentioned before, we're going to neutralize that stomach acid. Crazy cool thing about the small intestine. It's 7 meters long. A meter is slightly more than a yard. So this is crazy long, only about 30 millimeters wide. But if we were to flatten out the whole entirety of the inside of our small intestine, it would be equal to about a tennis court. It's a very large surface area inside that amazing organ. A couple little things that help the small intestine do its job. So that liver, as I mentioned before, and the gallbladder the liver is going to make the bile. The gallbladder is going to store the bile until we need it. Bile is made up of salt and bilirubin, which is formed from the breakdown of hemoglobin, and then some cholesterol. And this is just going to help to emulsify those fats, make it easier for our enzymes to get to them. The pancreas is going to make a bunch of enzymes, proteases, like endopeptidase, which is going to digest those proteins, lipases. Lipase is going to digest our lipids. Amylase is going to break down some starch. And we also have that bicarbonate that's going to neutralize that stomach acid, all made by the pancreas. So this is villi inside the small intestine. Just a quick reminder that we've got those microvilli on the villi. Those microvilli are just going to tiny little projections on those epithelial cells on the inside of the small intestine, increasing that surface area, making it easier for us to absorb lots of food. Once we get that food absorbed through the microvilli and the villi, some of our nutrients are going to go through the capillary network, part of the blood system. Fats are going to go into the lacteal because we don't want those fats clogging up those capillaries. Goblet cells are going to make mucus because you can imagine that if I've got chunks of food sliding through here all the time, we're going to dig up these cells pretty fast. So lots of mucus is made just to protect the small intestine wall from all that stuff going through the small intestine. Despite the very hard work of our goblet cells, we do actually replace the inside of our small intestine just about every 3 days. Crazy sauce. So I mentioned that we have lots of absorption happening across the wall of the small intestine, getting those nutrients out of the inside of the small intestine and into either our blood system or into those lacteals. A couple different ways that we can transport those nutrients into the small intestine. It can be through passive transport, no ATP required. We're always going from high concentration to low concentration. Examples of things that we transport passively: some monosaccharides, some vitamins, some minerals are going to go into the small intestine via facilitated diffusions. We either have some protein channels or protein carriers. Triglycerides, those fats, can actually pass right through the cell walls of the cells of the small intestine, and that's going to be simple diffusion. We also have some active transport happening, which requires energy in the form of ATP, where we're actually moving things against the concentration gradient, or we have big things that can't just get through the membrane. An example of active transport of nutrients is glucose, and we actually have some secondary active transport, which means this: we're actually going to use some ATP to pump some sodium out of the cells of the small intestine. That sodium now has a high concentration out here, a low concentration on the inside, so the sodium wants to go back in. But this protein that's going to let the sodium in is only going to let the sodium in if it brings the glucose with it. So we have this idea of a symport, co-transport, secondary active transport. We also have some bulk transport happening where it's not going through the membrane, but we're now going to enclose it in vesicles of the membrane. Exocytosis, we're spitting stuff out. That's what's happening here. The secretory vesicle is going to travel, dock to the plasma membrane of the cell, and then they fuse and the contents just are released into the environment outside the cell. This is an amazing micrograph image showing that happening. This happens often in our nerve cells. Neurotransmitters are going to be released into the synapse between 2 nerve cells via exocytosis. The opposite of that is endocytosis, and endocytosis is clearly the opposite, where we're sucking things in. So instead of releasing things out, we're pulling things into vesicles and then bringing them into the cell instead of letting them go outside. So those lipids in the bloodstream, I mentioned that they start off in the lacteal, but sometimes they do need to also get transported through the blood system. But those lipids, because they are hydrophobic, do not do very well in blood, since plasma is mostly water. And so what we are going to do is actually bind up those lipids, triglycerides, whatever else it is that we are transporting. We're going to bind those up into this lipoprotein complex. What's super cool about the lipoprotein complex, it is literally lipos. So we've got some lipids here, some phospholipids, but it's not a phospholipid bilayer, it's a monolayer. And so I've got hydrophilic on the outside, hydrophobic on the inside. And all of these triglycerides inside are also hydrophobic, and so they're super happy to hang out next to the fatty acid tails of those phospholipids. We're also going to have some proteins here that are just going to help to dock and transport these bubbles of lipids around. So lipoprotein complex is how we transport lipids like triglycerides through the bloodstream when they're not in the lacteals. We are almost done, my friends. Good work holding on. So the large intestine, the last little chunk of the digestive system we're going to talk about today. So the large intestine is all about water absorption. We might have a tiny little bit of final carbohydrate digestion. We've also got lots of E. coli bacteria in our large intestine. That E. coli is really good at making some vitamin K for it. Here's an amazing thing. We've got just about 4 pounds of bacteria in and on our bodies. There are 10 times more bacteria cells on us than our own cells, but they're way, way smaller. And so even though the bacteria cells are greater in number, our own cells are actually greater in like mass and biomass. But it's a crazy thing to think about, that we're 10 times more bacteria cell than we are human cell. Crazy sauce. And then one more piece of vocabulary words. So we talk about ingestion, getting food into our bodies. Digestion, breaking down the food inside our bodies, and then egestion is the excretion of that undigested food like cellulose. If we were eating bones or fur like some of our carnivorous mammal friends, we would also be egesting those pieces. Lots of bacteria also being egested from our large intestine through the rectum and then eventually out the anus. And that was a great awkward word to finish our lecture on. Good job today, my friends. Digestion review accomplished. We talked about metabolism, we talked about biochemistry, we looked at structure and function of the digestive system, organs, with that big focus on the small intestine. Keep up the good work.